respected dignitaries, faculty members, dear colleagues, and friends. On behalf of the Division of Genetics, Department of Pediatrics at AIIMS New Delhi, and the Institute of Medical Genetics and Genomics at Sir Gangaram Hospital, New Delhi, I extend a warm welcome to everyone. We are gathered here for an oration meeting in honor of Dr. Ishwar Chandra Verma, whose contribution as a mentor and a role model continues to inspire us all. It is a privilege to begin this academic session dedicated to his legacy. Today's oration is not just a tribute to his academic excellence, but a celebration of the values he has inculcated in us, the curiosity, the compassion, and the commitment to excellence. Some mentors teach us knowledge, some teach us skills, but a rare few teach us how to think, how to question, and how to serve with humility. Dr. Verma belongs to this rare group. It is with deep respect and gratitude that we begin this oration meeting in his honor, celebrating a life devoted to learning, mentorship, and service. May I now invite our special guests, Professor Srinivas, the director of AIIMS. Okay. Madam Dr. Shivali, maybe she's also on the way. Sir Dr. N.K. Mehra, to just say a few words. Dr. N.K. Mehra is the Emeritus Scientist at ICMR, and he's also a former Dean and the National President of the National Immunology and Transplant Society. Professor Mehra, please.
Thank you very much. We are here for the first Professor I.C. Verma oration. Professor I.C. Verma has been a colleague for us for many, many years. I joined the institute in 1969 as a student, of course, and I came from Amritsar. Dr. Verma was also from Amritsar. So we spoke the same language. I think our thinking was also similar kind. So very quickly we became friends. Of course, he was much senior to us, but very friendly, always very, very friendly. And my recollection of his moving around in the corridors of the institute in the early 70s was I found him a man in a hurry. He was always running around as if he wants to achieve something in the shortest time. And that was a goal. I think he was already thinking in terms of establishing genetics, clinical genetics as a component of the Department of Pediatrics. That department at that time, as I remember in the early 70s, and my wife, Raja, and of course, somebody is here, was a very dynamic department. Dr. O.P. Ghai as the head of the department, Dr. Mehban Singh, R.N. Srivastava, you name it, the people who were well known in their own fields. And then came the next generation, who were like my own peers, Maharaj Bhan and Veena Kalra and others. But everybody was getting specialized into their own field. You know, R.N. Srivastava wanted to do nephrology, pediatric nephrology, which is something that nobody at that time had thought about it. Vimalesh Seth wanted to look into juvenile arthritis and all. Dr. Ghai was, of course, well known in his own field of ICC. And Dr. Verma was very keen to establish genetics at a time when genetic diseases were not very that well known, and their diagnosis was becoming an issue. So his major goal was to establish laboratories where affordable diagnostics can be offered. And of course, started with thalassemia, beta thalassemia, deafness, and all. These are some of his issues that I do remember. Mental retardation in the children, and then took him to all kinds of genetic diseases and all. I always enjoyed interacting with him, and I was at that time establishing immunogenetics, like transplant immunology and immunogenetics. Another of the stalwarts has come, BBP Chaudhary, who also started his career from the Department of Pediatrics, but then went on to specialize in hematology. Uh, so, that department at that time, it's not just true of pediatrics, but every department. I would say in the 70s and 80s, even now, I'm sure now, because now that we have left the institute for more than a decade, but those were the formative years of the institute. And every department had stalwarts, and people used to take their names with a lot of respect and all. Um, Dr. I.C. Verma used to always interact with me. I said, look, I'm trying to establish transplant immunology and immunogenetics because the organ transplant were not happening, at that time. I remember in 1975, Dr. I.K. Dhawan and K.K. Malhotra came to me because they looked into me, somebody who has this interest, and they said, we want to start. But I got a lot of support from Dr. Verma. Later on, whenever I used to give talks on, we did some studies on the genetic diversity of HLA in India. He was very keen to see that. He always came to me lectures. After the lectures, he will mark some slides and he will call me in the evening. Yeah, send me those slides. That is really remarkable on the diversity of HLA B27, diversity of HLA2, and all that. His mind was always working towards genetics. How does the genetic diversity happen? Not just in these genes, but any other genes and all. And then he did establish the Department of Medical Genetics at that time. But we were together, I remember, in the meeting in ICMR, where ICMR was thinking in terms of developing this specialty even more, and a lot of people suggested rather than calling it medical genetics, we should call it as a genetic medicine, other way. And people used to use interchangeably words like genetics, genomics, and all. I think the correct word is genetic medicine. And he was the first, I would say, the first person uh, clinically looking into the genetic medicine. He really established that. Not only in AIIMS, he then went on to establish in Gangaram Hospital. Highly respected man all over the world. And I think we have enjoyed, myself and my wife have enjoyed a lot of love and affection from this couple. Kusumji always called me as brother, younger brother. And they not only just called as a brother, but treated us absolutely like that. And so when Madhulika wanted to organize this particular first oration, I said, I will definitely come, and I will come in person, because that's the kind of love and affection we have for this man who brought the subject of genetic medicine in India. Thank you very much.
Thank you, sir. Thank you very much. So now I call upon Professor Shifali Kulati, ma'am, who is the acting head of the pediatrics at AIIMS, to just say a few words, ma'am.
Morning, everybody. Respected ma'am. Professor Verma has been a professional par excellence, and I think there have been nobody in comparison to him I have ever known in institute. And so from the besides from the Department of Pediatrics, everybody has very cherished memories for him. And for me personally, he had been a great mentor. And I remember the first day I joined Unit 2 in pediatrics and his rounds, and each and everything which he taught, and I till date tell my students with his reference, and checking the treatment charts of at every bedside. And so and he used to also say that every day you check, and when you'll ask a question, he wants an fast answer, because his IQ was supreme, is the super genius, so he will not wait for somebody to respond. So I think that was really, really amazing. And what I remember, because I have a problem of speaking very fast, so he said, "Acha, tu meri tarah hi fast bolti hai." So I took it as a great compliment, and he's always held all of us hands, and I especially personally, I have indebted to him for everything during the beginning as resident to then becoming faculty. And I think the entire department is indebted to him for starting genetics, which now everybody sees in all systems and the way forward. And he thought it at a time, as sir pointed out, that nobody had thought of. So I think and it's very important to cherish his positive things and his, intellect. And I really feel, I last year, just before his demise, he was supposed to have given the Association of Child Urology India Oration. And that time, I remember you had come from the US, and I was wondering why sir is not responding to my emails. And then suddenly, ma'am responds on sir's mail. And then I talked to Ratna, and when Dr. Ratna went and spoke to him, so he was just said something in a complimenting way. So I really feel in that till the something he was privileged was till the end, he was contributing to science and also advocacy, because he's the way he handled parents of the patients, because that was something to cherish. We welcome you all for this oration, and we are very privileged to have it in AIIMS. Thank you so much.
Thank you so much, ma'am. I think we'll just go ahead and have the lamp lighting now. May I please invite
Please ask Dr. B.P.
Dr. B.P. Chaudhary, sir, would you like to say a few words? Sure, sir.
ये नए लोग हैं, इनको मालूम नहीं है, तो
नहीं,
very important.
sir.
Very good morning to all of you, particularly to Madam Verma. My association with Professor Verma has been very, very long. When I joined the Department of Pediatrics, I had just joined a year before, and we brought through the work together. Most important is he was very humane in nature, very much concerned about the children. He will always say, let us do best what we can do for them. They are the future of the country. And many times, since he developed a department, today is a department of genetics all over the country, he was a pioneer, he had a vision, he was a visionary. He said, let me do something which nobody else can do it. And patients used to come as a genetic disorders. They'll ask him, what will happen? How do we recover? He says, that यही तो मैं पता कर रहा हूँ कि आप कैसे ठीक होंगे. I mean, that was his real touch and consoling the parents, because for many congenital disorders, genetic one, treatment at that time was not available. And he was so much touched for their care. Some, I will not name the disorder. He made an association of that genetic disorder. He says, I cannot go to the government. Let this association can move to the government. And he supported them and finally the government gave the grants and for their welfare of it. And not for one, but several. He laid the foundation. He went to Jipmer also and worked. He worked in the various fields in the community for genetic disorders. And that was his humane. My association became much stronger besides being in pediatrics. As Professor Ghai said, everybody should have one specialty developed. And I took hematology, and we had a lot of genetic disorders, like commoner being the thalassemia, sickle cell, hemophilia. And so we had a very long association work together, and I used to interact with him to his office. He had a very remarkable memory. You go to his office, he will handle 2-3 people. I enter, he's talking to some, you just hold on, let me talk to him. He has to go and see the patient. He'll spend 10-15 minutes time. Somebody else walks in, he says, Chaudhary, it's an emergency, just wait for another. So he could handle. Some person from the lab will come. That was his capability and reacting with the people. The binary have always key how I could help. And that is where he worked. And when he even, after superannuating from AIIMS, he said, no, I have to continue and see the genetic department to develop. And that is how He spent rest of his life at Gangaram Hospital. Other is what he did was Indian Journal of Pediatrics. Now the journal which was almost, I say, submerging, almost had gone down, and he was handed over only the name of the journal and nothing. And from there, he brought this journal, which has the maximum impact factor in this country of all the journals except IGMR. That is the, and today everybody respects the Indian Journal of Pediatrics, for which he has been starting from the beginning and continued on this. And I was associated working for the journal with him right from the day one when it came. And we know the prints used to come, we used to correct, and next day it had to go to the press for correction and so on. He says, let us see that this survives. So his was beside genetics, beside care for general pediatrics. And then how could I develop pediatrics in the country? Education. And that is what he brought through, beside being a professor working through was the Indian Journal of Pediatrics. And I think that today person who is giving oration is again, I think, a committee who had selected his birth giving, have delivering it, because the genetics has as a basis for the cancer also. And we know today that a person who develops cancer has genetic susceptibility to go on into that, and I'm sure he's going to enlighten us and give us much more. I think the Professor Verma was a genius, par excellence, and a person the pediatricians all over the country will respect him for centuries to come. And it is always said, success of any person behind is the lady, and that is Mrs. Kusum Verma, who have been supporting her right and left. Even for journal, she gave money from her pocket so that it could survive the, budding part of it, journal in this department of pediatrics at that time. I think with this, I have all my respect. My pray for the Dr. Verma, and I hope his soul remains in the heaven and he blesses us from there to all of us. Thank you.
Thank you so much, sir. We will all now gather for the inaugural, for the lamp lighting. May I please request our chief guest, Professor Majumder, Madam Dr. Kusum Verma, Dr. Mehra, Dr. Chaudhary, Dr. Madhulika, Dr. Ninja, Dr. Ratna, Dr. Shipali ma'am. invite Madhulika ma'am, Professor Madhulika Kabra ma'am, to come and address the audience.
Thank you, Sunita. Good morning, everyone. It's my great honor, and I'm really humbled to introduce my mentor, Professor I.C. Verma, and a little bit about this oration as well. Dr. I.C. Verma was born on December 25th, 1936, in Kenya. He obtained his MBBS degree in 1961 from Punjab University, as Dr. Mehra just said, followed by MRCP from the Royal College of Physicians, London, and DCH from the Royal College of Physicians and Surgeons, Glasgow. Over the course of his distinguished career, he was awarded several prestigious fellowships and memberships, including MNAMS, FAMS, FRCP, FAAP, MACMG, and FIAP. Sir served for more than 3 decades as a faculty in the Department of Pediatrics at the All India Institute of Medical Sciences, New Delhi, from 67 to 1997. After superannuating from AIIMS, he founded the Institute of Genetics and Genomics at Sir Gangaram Hospital, where he served as director until 2017 and subsequently as advisor till 24. Respectfully remembered as father of medical genetics in India, he mentored many clinicians in the field. A visionary in the field, he played a pivotal role in establishing and advancing medical genetics in India, or genetic medicine, as Dr. Mehra said, maybe more appropriate word. Throughout his career, Sir received numerous awards in recognition of his remarkable contributions to research, academics, community research, outreach, and clinical services. So all his efforts were prone towards welfare of the patients and parents and how clinical genetic services can be improved in India. He served as a president of Society of Fetal Medicine, the Indian Society of Inborn Errors of Metabolism, and the Indian Academy of Pediatrics Delhi branch. As editor-in-chief of the Indian Journal of Pediatrics, uh, he actually did a great work in the area of pediatric education in addition to the genetics education. Later, he served as editor emeritus for the same journal. He helped shape the growth of one of the Asia's oldest specialty journals. So IJP is the oldest specialty Asian journal in pediatrics. Dr. Verma worked tirelessly for the recognition and management of rare diseases in India and made significant contributions to the development of national guidelines and policy for the treatment of rare diseases. And as you now know, we have a national rare disease policy existing in India. In acknowledgement of his lifelong services, he was conferred the Padma Shri by the Government of India in 2023. To honor his extraordinary legacy, the Dr. Ishwar Chandra Verma Oration has been established as an annual event beginning this year only, that is 2025, jointly organized by the Division of Genetics, Department of Pediatrics, AIIMS, New Delhi, and the Institute of Genetics and Genomics, Sir Gangaram Hospital. The oration will be held alternately at the two institutions every year. This annual event is dedicated to celebrate Sir's inspiring journey and his immeasurable contributions to patient care, research, and in the field of medical genetics. Thank you very much.
Thank you, ma'am. Now, may I please invite Dr. Ratnapuri, ma'am, to read the citation.
Thank you very much, Sunita. I have the pleasure to read the citation for the first Professor Ishwar Chandra Verma Memorial Oration for Excellence in Genetics and Genomics 2025. Dr. Partha P. Majumder, Distinguished Professor of the John C. Martin Center for Liver Research and Innovations, Kolkata, and Emeritus Professor, Indian Statistical Institute, Kolkata. He has been the National Science Chair in India and has significant contributions to human statistical and population genetics and genomics. He has founded the National Institute of Biomedical Genomics, India. Dr. Majumder has developed methodologies for mapping human disease genes, identified genomic factors, underlying many diseases, notably oral cancer, and has reconstructed the ancestries and relationships of ethnic population groups of India and Asia using genomic methods. His contribution to capacity building in human and statistical genetics in India are immense. He is an elected fellow of all the three National Science Academies of India, the World Academy of Sciences, and the International Statistical Institute. He has served as the president of the Indian Academy of Sciences and of the West Bengal Academy of Science and Technology. He is a member of the boards of the Human Genome Organization and the Human Cell Atlas. He serves as an expert on genomics for the World Health Organization. Dr. Majumder has been honored with the G.N. Ramachandran Gold Medal 2021 of the Government of India, the Barclay Memorial Medal 2020 of the Asiatic Society, Golden Jubilee Commemoration Medal 2018 of the Indian National Science Academy, and Biology Prize of the World Academy of Sciences Trustee 2009. To honor the legacy of Professor Ishwar Chander Verma, the Division of Genetics, Department of Pediatrics, AIIMS, New Delhi, and the Institute of Genetics and Genomics, Sir Gangaram Hospital, New Delhi, are privileged to confer the first Professor Ishwar Chander Verma Memorial Oration for the year 2025 to Dr. Majumdar for his exceptional contributions to the field of human population and statistical genetics and genomics. We request Dr. Majumdar to please deliver the oration. Thank you.
I'm absolutely honored and thrilled to be called upon to deliver Ishwar Verma Memorial Lecture. It's indeed a pleasure for me because Madam Verma is here, my friend Narinder Mehra is here, Shinjini Bhatnagar is here, and actually I've known Madhulika for a very long time. I set foot on the soil of All India Institute of Medical Sciences with Dr. Verma holding my hands. Showing me the little building where the medical genetics department was coming up, and Madhulika had just joined at that time. So it's been many years. I actually met Dr. Verma either in December 1972 or in January 1973 in the second annual conference of the Indian Society of Human Genetics, which was being held at that time in Bombay, as it used to be called, Mumbai now. That's when I met him, and ever since then I've been, closely associated with him in many ways, mostly on the domain of human genetics and on the turf of the Indian Society of Human Genetics, of which he became a president at a later point of time. The second annual conference was in Mumbai when I met him, and yesterday, I actually conveyed most of the people who actually founded the Indian Society of Medical Genetics. They are no more with us. There is one person who is still living. His name is Kailash Malhotra. Some of you may know him. I actually wrote to him yesterday that I'm going to deliver this talk, and this morning he responded saying, "Wow!" and he remembered Professor I.C. Verma as well. So Professor Verma actually made a sort of established human genetics and of course medical genetics in India, and we all know that. It's really been my pleasure knowing him. Also want to recall one other thing. I don't want to recall too much because I'll become very sentimental. At a time when the government of India was seeking to build new institutions, and they asked various people in the country to provide names of new institutions or ideas for new institutions, I was called by Dr. Bhan, and I provided an idea that started as a few lines to begin with and eventually became an institute all by own. At the same point of time, there were other institutes. There was one other institute, actually two other institutes that were coming up. The Translational Health Institute and the Regional Center for Biotechnology. What I was called upon to deliver, deliver to the nation, for which I had given the idea, was to form this National Institute of Biomedical Genomics. And in doing this, we went through a very long series of consultations, and Professor Verma was one of the biggest supporters in the formation of this National Institute. He visited the institute. As a matter of fact, on the first foundation day, our institute was not even, not even one brick was laid. We held it in a place called Manovikas Kendra, and he actually came and delivered a talk on Down syndrome, which was the centenary of the Down syndrome. So it was, I've been associated with him in many, many ways, and it's really my great pleasure and honor to be able to deliver today's talk. I've, I need to be able to, control this. Okay. All right. So this, all right. So, yes. So as you can see that this is Professor Verma. He is giving a talk. He's giving a talk on human genetics in India. And this is what he said. And I was there for this talk. It was fairly late. National Institute of Biomedical Genomics was being formed. He said that O.S. Reddy developed the first human genetics unit in India in Osmania University, Hyderabad, and this is what India looked like essentially to begin with. And he said that at that time it was only humans and no genetics. There was no genetics at that time, and this is what he emphasized. This is 1968. And then he went on to say that, today we have the National Institute of Biomedical Genomics, and he was one of the greatest supporters of this institute that I founded. And, well, it's not in Kolkata, but close to Kolkata in a place called Kalyani. And he mentioned something in his talk. Again, this is called ICGP. Actually, it should have been ICGC, International Cancer Genome Consortium. And this is what I'm going to talk to you about, the findings of the International Cancer Genome Consortium, which the government of India had participated in. And there were two principal investigators, one a clinician, one a genome scientist. The clinician was Rajiv Sareen from Tata Memorial Hospital in Mumbai, ACTREC, Director ACTREC, and I was the person who was doing genomics. So what I'm going to describe to you today is essentially that. So I'm going to talk about evolution of the cancer genome. If genomics has had any major impact on any complex disease, it is cancer for sure. Nobody is going to dispute the fact that many of the treatments that we provide for cancer today are essentially anchored on genomic findings. It's a disease of the genome, and that's at the end of my talk. I hope that I'll be able to convince you that it is truly a disease of the genome. When we talk about a disease of the genome, we think that it should run in families and it should be a hereditary disease. But cancer is not a hereditary disease. Only about 10% of cancer of any organ will be familial. It will run in families. It will run from one generation to another. The remaining will be non-familial, and yet we are calling cancer the disease of the genome. If you look at, for example, this is from breast cancer, and if you look at breast cancer, that's what you see. I mean, this is one of the most aggregated families of breast cancer, where you have lots of people afflicted with breast cancer, yet it truly does not run in families. In this particular pedigree, it does, but you only see about 10% of breast cancers to be familial of this nature. So why are we calling this a disease of the genome, and when in fact it is non-familial? That's because we have, genome is split into two parts, so to say. One is the germline genome and the other is the somatic genome. When there are alterations in the germline genome, it will, of course, run in families. That alteration will run in families, but when it's in the somatic genome, it will arise in that individual. The alterations will be there in that individual. Whatever phenotypic effects those alterations will impact on that individual, but will never be passed on. This is what most of the cancer, yet it's happening in the genome. So it is definitely alterations of the genome. So let me explain this a little bit more, and why. My background is in statistics. Over a period of time, I've gotten into genetics, and why a statistician should become interested in genetics and what role does a statistician have to play in genetics is best exemplified when we deal with these complex diseases of the somatic genome. And I will present to you what the complexities are. So imagine, if you will, that this is the epithelial layer, and these are all cells that are stacked below the epithelial layer. This is how the cells look when we are born. These are all somatic cells, not germline, somatic cells. Over a period of time, as we gain age, there are alterations in the somatic cells. And so those are mutations that have arisen in the somatic cells. And these mutations are happening all the time, even as I speak. Perhaps somatic mutations are happening in my lung cells. So this happens all the time, and we're completely oblivious to it. We don't care about them, except that sometimes these cells do something. They don't use these mutations or alterations do something to the cell that provides the cell a growth advantage. Usually our cells will divide from 1 to 2 to 4. 2 to the power of n is how they divide. But these cells sometimes violate that because of this alteration, and more mutations arise. And as a result of that second mutation, this particular cell got a growth advantage and grew 3 more cells as opposed to growing one to 2. So this mutation, this alteration gave the cell a growth advantage. These are happening all the time, and we again don't care about them. We don't get to know them. That particular mutation also gave this cell a growth advantage. And on the background of these new the cells that have already gotten alterations, new alterations can arise. So, for example, in this particular cell, which is clonally propagated as a result of that yellow mutation there, a red mutation has arisen. So this red mutation again might or might not do anything to the cell, except that this red mutation does to the cell what the previous mutations did not do as much. Provides the cell a huge growth advantage. It breaches the epithelial layer and actually becomes a malignant tumor. So when we are looking at these different kinds of genetic alterations in these cells, somatic genetic alterations in these cells, this red alteration behaves very differently from the previous alterations, and we care about these red alterations because that red alteration on the background of the black and blue provides the cell this huge growth advantage. So we need to know what are those alterations that provides the cell a huge growth advantage such that it becomes a malignant tumor. So this is what we care about. Even after this particular alteration has arisen, there are alterations that are happening. So for example, these green alterations are happening, and so on and so forth. So this is, and what we really need to know is what are the genes and the genomic alterations that drives the cell to becoming a malignant tumor, that is, provides the cells a huge growth advantage. This is what we need to know. So if we can identify those genomic alterations and the genes in which those alterations arise, then probably we'll be able to understand the disease better. And if we understand the disease better, perhaps we can control the impact of those genetic alterations and treat the cancer that we care about. So this is the reason why we call cancer, why we talk about the cancer genome, and that's the evolution, because it's not happening in one day. It's happening over a period of time as we gain age. These somatic alterations arise, and so usually it involves more than one mutation. It usually involves multiple mutations. Did we not know this? Why is it that we are now talking about cancer and cancer genomes and so on and so forth? This has happened mostly in the last 10 or 15 years. But was the knowledge not there? Of course, the knowledge was there. Did we not know how to dissect these cancer genomes? Of course, we knew how to dissect. Did we have the wherewithal to dissect? We didn't have the wherewithal to dissect. So this is a Nobel laureate, Renato Dulbecco, who actually said, if we wish to learn more about cancer, we must concentrate on the cellular genome. We have two options: either to try to discover the genes important in malignancy by a piecemeal approach, which is how I started my life, which is where Professor Verma started his life, looking at these genes important in malignancy or other kinds of human genetic diseases by a piecemeal approach. This is what we did, one mutation at a time. Or to sequence the whole genome. This is what we do now, when we thought about this, this is 1986, it was a paper published in Science, how to sequence the whole genome, the wherewithal didn't exist. We didn't have machines that could actually not even conceptually how we would sequence the whole genome. And he went on to say it will be far more useful to begin by sequencing the cellular genome. Of course, so we knew that sequencing the cellular genome would be good for us to be able to identify the genes and genetic alterations that underlie cancer, but we just did not have the way to do it. And the way to do it came about in the last 15 years, and so we are now able to run. I'm going to focus on oral cancer primarily because that was India's contribution to the International Cancer Genome Consortium. And the reason why we chose oral cancer is because India has a very high burden of oral cancer, and this burden is primarily among the males. We actually proposed to the government of India that we do two cancers. The one that's the most prevalent among males in India, which is oral cancer, and the one that's most prevalent among females in India, which is cervical cancer. But these were expensive propositions at that time. Now the costs have come down, but when we did initiated all of this work, it was very expensive, and the government of India said we cannot afford, we cannot provide so much money to the International Cancer Genome Consortium's India project. So if you look at the other interest in it's not like we didn't want to look at the female cancer. It's because we had the cervical cancer is primarily because of infection by the human papillomavirus. That's the causative agent. We know that. Yet there are host, pathogen interactions, etc., which are of great interest. It's not like we wanted to ignore the females. It's because so oral distribution also provided us with a scientific background because of its site distribution. If you look at the West, and by the time the International Cancer Genome Consortium started, the United States had already started a project called the Cancer Genome Atlas, TCGA. And there they were also looking at oral cancer. They were looking at a variety of cancers that are prevalent in the United States. They were also looking at oral cancer. If you look at oral cancer in the US, about 70-75% of oral cancers are on the tongue or the floor of mouth. And only about 25% of the cancers are in the remaining part of the buccal cavity. If you look at India, only about 25% of oral cancers are in the tongue or the floor of mouth, and the remaining are in the remaining areas of the buccal cavity called the gingivobuccal cancer region. So what we said is that if we look at the Indian oral cancer and focus our attention to the gingivobuccal region, which is really excluding the tongue, we will be able to compare our results with the results of the United States Cancer Genome Atlas data and see what differences are there. So we'll be able to build the entire story of oral cancer, both the tongue and the non-tongue oral cancer. So that's the reason why this particular cancer type was chosen. And like I said, Rajiv Sareen was the person who was the clinical person who generated the clinical data, provided us with the samples that we needed to analyze from a genetic point of view. And over a period of time, we have now also started collaborations with various other clinicians. Anil De Cruz was at the Tata Memorial Hospital at that time. P. Arun right now is in Tata Medical Center in Calcutta, and Sandeep Ghosh is in the R. Ahmed Dental College in Calcutta. I'll have a little bit to say about our collaboration with Dr. Ghosh. And the two major pillars with whom I have been working is Arindam Maitra in the National Institute of Biomedical Genomics. He manages the experimental setup, and Nidhan Biswas, who manages the data analysis setup. And so they have been the two major pillars because of which we have successfully been able to complete the Indian commitment to the International Cancer Genome Consortium project. The International Cancer Genome Consortium project had lots of different kinds of cancers, and we were working in oral cancer, and that's India, and none of the other countries was working on oral cancer. And the goal was to obtain a comprehensive description of genomic, transcriptomic, and epigenomic changes in 50 different cancer types or subtypes, which are of clinical and societal importance across the globe. Our sample size was 500. We calculated the sample size that was necessary to identify all of the important genomic alterations. The number of patients was 500. Most patients were male. 85% of the patients were male. Most patients are young. Most patients are tobacco users, and of course that's known. That's one of the major driving forces, environmental drivers that creates these genomic alterations. Most of our patients were tobacco users. There was no significant HPV involvement. I bring this up primarily because tongue cancers have a significant HPV involvement, as gleaned from the data of the Cancer Genome Atlas project in the United States. I won't get into the gory details of this. So we sampled these patients and obtained samples from them, and I'll describe what the samples are. This is the hazardous combination. The snakehead line chews up the epithelial layer. The carcinogens in the tobacco then hit the cells directly, and that's what promotes cancer. Before I come to this, I just want to make a mention of this, that when we are talking about these 500 patients, what are the biospecimens we are gathering from them, and what are the biospecimens that we are analyzing? We are generating data or sampling DNA by extracting from the cells of the tumor tissue and also of the normal tissue. And the reason why we also need to sample the normal tissue we usually will have mostly germline mutations, and there are many germline mutations. The tumor tissue will be a combination of germline mutations and somatic mutations, the cells in the tumor tissue. We don't want to touch the germline mutations because our major focus is to identify the somatic drivers of. So what we do is we sequence both, and whatever changes that we have in the germline from the somatic cancer genome, we subtract those, so to say, so that we are left with only the somatic drivers, and then we do the statistical analysis on the somatic drivers to identify, on the somatic mutations or alterations to identify which ones are driving the cells. This is it involves so there is a lot of background noise meaning that lots of alterations that really don't mean anything to us because they are not the drivers, but they happen in the background, and so we need to get rid of them, or in other words, we need to identify the signal from the noise, and that's a lot of statistical methodology, and I'm not going to get into that. What we did was, after sampling these patients and doing the statistical analysis, we identified that there are 10 genes that are drivers of gingivobuccal oral cancer, and the percentage of patients with mutations are provided by these bars and different kinds of genomic alterations are also marked, but we don't get into that today or right now. If you can't read the names of the genes, these are the names of the genes that we found were drivers, or alterations in those genes were drivers of oral cancer. And so the next question was, okay, so we found these 10 genes. We know that cancer genes behave in two ways, or belong to two classes of genes. One's called tumor suppressor, the other is called oncogenes. The tumor suppressors are awake most of the time. They are suppressing the tumors, they are keeping tumors in check. And the oncogenes are sleeping giants, they are sleeping all the time, and the genomic alterations wakes them up, and they start proliferating, providing the cells a growth advantage, and those cells become malignant tumor. So the question that we asked is, are these genes, and some of these genes can behave both as tumor suppressors and as oncogenes. So the question that we asked is, are these tumor suppressors or oncogenes? How are they behaving? So we went ahead and did that analysis, and like I said, that tumor suppressors, they are usually awake. Genomic alterations, or some of these genomic alterations, will make them go to sleep. And once they go to sleep, they will no longer be able to do their normal activity. And so those cells, when there is a further alteration, they may gain a growth advantage, etc. And the way that we do it using genomic data is that if you look at a tumor suppressor gene, when there are alterations, you will find alterations strewn all over the gene, and these heights of the bars are the number of patients in which there is a mutation in that particular that particular spot in the gene. So the genomic alterations are strewn all over the genes, as opposed to oncogenes, which are sleeping giants, and they will wake up when there is a genomic alterations. But those genomic alterations have to take place in particular places, and so the number of alterations that you find in an oncogene will be small. And so the take-home message from here is that if a cancer type involves tumor suppressor genes, because alterations can happen anywhere in the gene to suppress the activity of the tumor suppressor, it will be very, very difficult to act upon tumor suppressor genes from a treatment point of view, from a therapeutic point of view. On the other hand, if the cancer is happening because of alterations in an oncogene, because the alterations will be in a fixed number of places and large number of patients will have those kinds of alterations, it's much easier from a therapeutic point of view. So it was of interest for us to find out whether the genes that we identify are serving as tumor suppressors, are acting as tumor suppressors, or as oncogenes. And to our dismay, what we found was that these genes are acting as tumor suppressors. P53, for example, can act both as a tumor suppressor and as an oncogene, but in the context of gingivobuccal oral cancer, it's working as a tumor suppressor, because that we can know this because of the distribution of genomic alterations in P53. So this was our major finding, and this is the finding that we originally published. If we, like I said, that the Cancer Genome Atlas also was working on head and neck squamous cell carcinomas, and their data comprised primarily from tongue and floor of mouth data. And when we compared our data with the data of the Cancer Genome Atlas, what we found was that there were 3 new genes that were involved in gingivobuccal oral cancer that were not present in the tongue and floor of mouth cancer. And of the genes that were common, and of course there are several genes that are common, of the various genes that were common, one particular gene stood out, caspase 8, which had a frequency of 43% alterations among gingivobuccal patients, while only 9% of tongue floor of mouth patients had alterations in that gene. So there were significant differences in frequencies of patients who had those kinds of alterations. So this was our finding with respect to oral cancer, frank oral cancer. We then wanted to understand, okay, these 10 genes are involved. It cannot be that these 10 genes are simultaneously getting altered. So there must be a progression of alteration from an early stage of cancer to the frank cancer stage, and can we map them? Can we map which gene is getting altered first, followed by which gene, etc.? We were not able to completely sequentially find that particular progression, but what we did find was that, well, as most of you would know, that from the normal you have a dysplastic change, then you have a hyperplastic change, and eventually frank cancer. This is called, usually these are seen as white patches in the mouth called leukoplakia, followed by what's called submucous fibrosis, and then frank cancer. And like we said, that these are 10 genes, but can we find out which gene is getting mutated first, followed by which gene, etc., so that if we can identify which gene is getting mutated first, we can manage the disease better, right? So that's the whole idea of looking at progression. And so let me just so we were lucky that we got a cover of the Journal of Pathology. For doing this, we had to find patients who had both leukoplakia and frank cancer in the same oral cavity. So we identified those patients are not easy to come by, primarily because when they have frank cancer, the leukoplakia is gone. The leukoplakia has transformed itself to frank cancer. So we actually had to go to a dental college, and that's where I mentioned our clinician who works in the dental hospital provided us with the sample. So we sampled 4 different kinds of tissues from each of these patients: blood, tumor, frank cancer, leukoplakia, and an adjacent normal tissue from within the oral cavity. And then we went ahead and did, I will skip all of the gory details. We went ahead and did all of the various kinds of sequencing, whole genome sequencing, and analyzing that data. We also, what we did was to recruit, sample sizes were not great. We recruited 11 individuals who had leukoplakia without any tumor in the oral cavity. That's because the reason why we wanted these samples is because there is a field cancerization effect, as many of you would know. So when you have frank oral cancer and leukoplakia in the same oral cavity, leukoplakia samples may, because of field cancerization, may get impacted on. So if we can look at leukoplakia sequences from within the oral cavity of a cancer patient and also see whether field cancerization takes place, if we can get leukoplakia samples from outside of patients who do not have leukoplakia in the oral cavity, who do not have frank cancer in the oral cavity. So we did all of this kinds of analysis, and again, let me just tell you the summary. We were able to, like I said, for all the 10 genes, we were not able to sequentially arrange them, but we without any major discussion about, if you look at the data, we can easily figure out that caspase 8 is the gene that gets knocked out first or altered first, followed by NOTCH1 and P53. The remaining genes, we couldn't actually arrange them in sequence, but there is EGFR amplification and chromosomal instability. Then there is tumor-specific inflammation and all of this. So at least we were able to figure out what are the first 3 genes that get altered as an oral cavity moves from normal to leukoplakia to oral submucous fibrosis and so on and so forth. So it's caspase 8. So this is very useful information in terms of management. When we generated these data, we also identified that there is a lot of heterogeneity between cancer patients, or even within a cancer patient. If you look at the cellular level, even within a cancer patient, and we looked at transcriptomic data and all different kinds of data, we were convinced that both intra and inter-tumor heterogeneity was evident from the data that we had generated. So what we did was to identify whether we can classify patients by into clinical subgroups by looking at the kind of heterogeneity that there is within their oral cavity, within cells of the oral cavity at the single cell level. Now, this is not something that's easy to do, primarily because you're now looking at one particular cell. We can't isolate DNA and start sequence that DNA because that ability still does not exist. But what we can do is to isolate RNA, mRNA from there, and be able to identify the transcriptome of that particular one cell. So if we sample from the oral cavity or from a tumor, let's say 100 cells, and we sequence each one of the cells, then we can identify the extent of heterogeneity in the transcription levels of these various cells and which genes are transcribed in these various cells. Are the same genes transcribed in the various cells within a patient or across patients, across oral cancer patients? Those were the kinds of questions that we asked, and what we did was to, this is very difficult proposition, so we were only able to get 12 treatment naive OSCCGB patients. Not that we don't have the ability to do it, but it's enormously expensive. It takes about 4 lakh rupees to look at about 2,000 cells from one individual and be able to generate the transcriptomic data. Anyway, so what we did was to harvest about 50,000 cells from these 12 treatment naive patients. We went through all of the different kinds of data curation, etc. We were able to eventually get about 30,000 cells. These 30,000 cells is a mixture of both is a mixture of both malignant and non-malignant cells. And so what we first did was to identify the malignant and non-malignant cells from here. So those were the non-malignant cells. This is a cluster of malignant cells. The non-malignant cells are all mixtures from different patients. The malignant cells, and I'll come to the malignant cells, which is really our interest. I'll skip this. So in doing so, we also devised a method by looking at transcriptomic data and identifying the malignant cells from the non-malignant cells, which is really a difficult chore. But anyway, so I'll skip this non-malignant cells. I'll focus on malignant cells. So we were able to get about 7,500 malignant cells, and you see these clusters of cells. These are cells that are derived from individual patients, and we can easily see that there are 2 groups of clusters, or clusters that belong to 2 groups. So we were wondering what these groups are. We went back to the patients from which these cells had arisen, and what we found was that, well, before that, most clusters were turned out to be patient specific. So these are various clusters, and if you look at these are patients, and if you look at clusters, for example, as you can see that this orange thing belongs to one single patient. So these clusters are mostly patient specific, in which case what it really means is that there is an enormous inter-tumor heterogeneity. Two patients with the same kind of cancer are not expressing the same set of genes at the same level. So there's an enormous inter-tumor heterogeneity. We were also wondering what these two groups are. Interestingly enough, all of these patients, all of these clusters essentially belong to three patients with oral submucous fibrosis, and the other cluster is from the remaining patients. So the oral submucous fibrosis patients actually stand apart. And please remember that as a patient goes from a normal tissue to a frank cancer tissue, not all of the patients will suffer from oral submucous fibrosis. So that's a special clinical entity. Okay. So we also asked ourselves, are there certain genes that can differentiate between patients with oral submucous fibrosis and those that don't go through this particular clinical stage? And we were able to identify three genes that alterations in which can predict with 100% certainty whether they have oral submucous fibrosis in the oral cavity or they do not. When we analyze these cells, what we identified again is that most of the specific clusters of cells that comprise about 41% of all malignant cells undergo epithelial to mesenchymal transition. Epithelial to mesenchymal transition is a hallmark of cellular changes in cancer of any kind, as a matter of fact. Bob Weinberg, just 3 days ago, gave a major talk on epithelial mesenchymal transition and the epigenomic factors that impact on epithelial to mesenchymal transition. That results, and this as a result of this transition, you get tumor invasion and metastasis. So we were able to identify those. There were two things that we identified, that there are certain clusters of cells that are involved in EMT, which results in metastasis and invasion. And also we were able to see that there are some, so these are clusters of patients with that have EMT or partial EMT. Not all of them, at the stage that we sampled, not all of them had expressed EMT, epithelial through mesenchymal transition. There are also fetal signatures. So there are fetal epithelial cells that are abundant in some of the malignant tumors. So eventually what we were able to do is to show that these 12 patients that we had sampled, there is an enormous amount of variation, heterogeneity in the genes that are expressing in the cells of the tumor tissue. And these 5 patients are expressing cells. 5 of these 12 patients were predominantly expressing cells that are cells are predominantly expressing genes that are involved in epithelial to mesenchymal transition. 6 of those patients were expressing genes that are that have fetal cell type signatures. So there are 2 mutually exclusive classes of patients that we can group. And the third is that makes 11. The one single patient probably had germline epithelial cells, and we are not very sure that particular patient could not be classified into either of these 2 clusters. So knowing that there are different kinds of clusters of patients is also helpful in identifying the extent of heterogeneity among patients with oral cancer, with gingival oral cancer. So overall, depending on whether I should stop here or go on, overall, so far what we have done is, or what I've been able to show you is that we identified genes that drives at the cellular level into oral cancer, and the nature of the function of these particular genes, which I actually didn't tell you much, except to say that these are tumor suppressors primarily. We identified the sequence of genes that are significantly altered as an oral tissue progresses from normal to precancerous stage, caspase 8, NOTCH1, TP53, etc. Identified that malignant cells that transit to 2 broad mutually exclusive states, the EMT and the fetal cell-like signature, each state being specific to a patient. All right. Do I have 10 more minutes? Okay. So if I have 10 more minutes, what I'm now going to show you is essentially dissect the molecular heterogeneity a little bit more. And the way that we are dissecting this molecular heterogeneity is stems from our initial feeling that there may be some changes in immunological genes, and immune therapy might be applicable to. That was the hypothesis that we began with. So we wanted to see if we can show that there are ways and means of using immune therapy in oral cancer, whether we can provide the evidence. So we started with 72 tumor patients, and we had adjacent normal from these patients, and also we had an independent set of 25 leukoplakia patients. We isolated mRNA and essentially did RNA sequencing to do transcriptomic analysis of these. The transcriptomic landscape of the OSCC GB patients, and the question that we asked is, what are the sets of genes that are significantly enriched in oral submucous oral squamous cell carcinoma of the gingivobuccal region? And again, I'm not going to go through all of this. So essentially, what I can provide you, the summary is that 33 hallmark gene sets are significantly enriched in tumor types. So once we generate the transcriptomic data, we can actually analyze that data to find hallmark gene sets that are involved in these transcription factors, these transcriptomic data. And what we found was that in tumors, there is positive enrichment in about 80% of the genes that are involved in activation of immune functions, angiogenesis, cell cycle related functions, TGF beta signaling, P53 pathway, and glycolysis. Negative enrichment was with respect to deactivation of some metabolic pathways like bile acid, fatty acid metabolic pathways, and oxidative phosphorylation. The one most important thing was for us the activation of immune functions. There was positive enrichment with respect to activation of immune functions. So it provided us with some indication that our hypothesis that immune therapy might actually work. We needed to provide more evidence. So we went ahead and using this transcriptomic data, what we were able to do is to show that of these 72 patients, 55 patients belong to one single cluster, and 17 patients belong to another cluster, which we call as GB1 and GB2. What are the major differences between these two clusters of patients? So compared to GB1, these GB2 clusters, which is a larger cluster, showed a positive enrichment of apoptosis, apical junction, and angiogenesis. And GB1 patient showed a higher enrichment of DNA repair and glycolysis. And JAK-STAT signaling pathway was positively enriched in all of the GB2 patients, but only in about 53% of the cluster with smaller number of patients. The abundances of abundances of immune cell types similar between clusters of tumors? The answer is no. And what we were able to show is that compared to GB1, these GB2 tumors, which is the larger fraction of patients with those kinds of tumors, they have significantly higher abundance of B cells, both memory and naive, M1 macrophages, not M2, monocytes, CD4 T cells, both activated and resting, and CD8 positive T cells. The implication of all of this is that we looked at immune checkpoint receptors, and we found that immune checkpoint receptors are involved. Similar pattern, this is CD8 positive T cells. Similar pattern was also found with CD4 positive T cells. So immune checkpoint receptors were active. We wanted to find out whether the ligands were also active, the PD1, PDL1 ligands, for example. And what we were able to show is that compared to GB1, the larger group of patients that belong to GB2, those tumors showed significantly higher expression of immune checkpoint ligands. So this is interesting that we have immune checkpoint receptors and the corresponding immune checkpoint ligands also that are involved. So there were significant positive correlations that we observed between immune checkpoint receptors and the corresponding immune checkpoint ligands. This is very, very useful from the point of view of understanding whether immune checkpoint therapy might work. So these kinds of correlations that are very highly significant are suggestive of an active immune checkpoint blockade mechanism. And what we have been able to show, again, very quickly summarizing, taken together, what we believe is that these GB2 tumors, which is the major class of tumors, are immunologically hot with higher T cell infiltrations, and therefore GB2 patients are more likely to benefit from immune checkpoint inhibitors compared to the GB1 patients. So we looked at for immune checkpoint therapy to work or to be adopted, we also need to show that as progression takes place, the same phenomenon happens, and we were able to show that as one moves from normal to leukoplakia to tumor, we do find exactly the same phenomenon. I won't get into the gory details. So there is monotonic upregulation of immune ligands in from normal to leukoplakia to tumor tissue, and that monotonic upregulation is very, very important for adoption of immune checkpoint therapy. So the question is, can we find the last question that we asked ourselves is that can we find biomarkers to differentiate between GB1 and GB2 clusters of OSCGB, because you don't want to adopt immune checkpoint therapy to all patients, because we did find that there are two clusters of patients. So and GB2 clusters of patients are more likely to benefit from immune checkpoint therapy. So early on, we need to, given the patient, we need to figure out whether that patient belongs to GB2 or GB1. So we said that can we have a genetic angle to discriminate between those two kinds of patients? And the answer is yes. There are three genes that most differentially express between GB1 and GB2, CD2, 26, 38, and KBT-BD8. These are the three genes, and when we use these three genes to classify patients, the percent of correct classification is 86%, which is really good. So essentially, then I will wrap up by saying that we've been able to identify genes, genomic alterations, the driver genes and the driver alterations. We've been able to figure out, what kind of progression is there. We've been able to figure out at the single cell level how much of heterogeneity. We use that data to classify patients into groups and ask ourselves, can immune checkpoint therapy work in oral cancer? And the answer to the last question is yes, not in all patients, but in the larger majority of patients. And we think that this work has come to a fairly reasonable conclusion. There are still some questions that we need to answer, and we are engaged in those kinds, or at least Nidhan, Biswas, and Arina Mitra are engaged in those kinds of questions. And so we hope that I hope that I've been able to provide to you a nearly wholesome picture of our research on oral cancer, and that it has gotten to a translational benefit as well. Thank you very much for your attention.
Thank you so much for such an amazing lecture and really guiding us through the whole process of cancer. May I now request Dr. Ratnapuri Madam, Dr. Madhulika Ma'am for the felicitations.
There are some questions. Usually they are not asked.
No, there are questions, but
So please,
usually they are not asked.
they are not asked.
ये मेरा दोस्त है. I didn't want to embarrass him, but I mean I will ask him during tea time.
Ma'am, either this side on YouTube पर आता नहीं है. YouTube पर नहीं नीचे. Sir, right. Okay. It's a scroll.
अब दीजिए.
Suneet पढ़ा. काम सुनता.
Thank you. It's such an honor. Thank you very much.
डबल हो जाएगा.
Thank you. With this, you look like a chancellor now.
Can we all come for a group photo here?
So now we have come to the end of session. A very good morning to one and all. It has been a privilege and honor to host the first IC Verma Memorial Oration today. On behalf of Division of Genetics, Department of Pediatrics, AIIMS New Delhi, and Institute of Genetics and Genomics, Sir Gangaram Hospital, Delhi, I thank Dr. Partha Majumder for sparing his time for this Professor I.C. Verma Memorial Oration. I would also like to thank both offline and online participants for being a part of this memorable event. And I thank SET Facility and CMET and AIIMS administration for all the arrangement. So I thank you all and I request everybody to kindly join us for tea and refreshments. Thank you.